COVID-19 continues to keep us on our toes. That's what we'll be talking about in this video. SARS-CoV-2 is the virus that causes COVID-19. It is a coronavirus, which is named for the crown-like appearance of their spike proteins, similar to the corona of the sun. SARS-CoV-2 is not the only coronavirus that causes illness in humans. Coronaviruses are known to infect both mammals and birds, where it often causes respiratory tract infections. Some coronaviruses cause things like common colds, whereas others can cause MERS, SARS, and also COVID-19. So SARS stands for Severe Acute Respiratory Syndrome and was an epidemic in 2002 through 2004 in China and several other countries. There were fewer infections than COVID-19, but the frequency of lethality was higher. So the SARS outbreak caused many of these countries, including China and some of its neighboring countries in Asia, to become aware of these pandemics and the importance of stopping the spread. Now, some of the information that we assumed would be true for COVID-19 because of SARS did not end up being true. And one of those was that in SARS, the disease was transmitted mostly when an individual was showing symptoms. But we saw this spread of COVID-19 before individuals started showing symptoms, or sometimes some didn't really seem to show symptoms at all, and that was different than we had seen for SARS. And again, that had informed some of our early responses to the pandemic that didn't end up quite being sufficient. Now, another past coronavirus was MERS, Middle Eastern Respiratory Syndrome. MERS is caused by MERS-CoV and has a 35% lethality rate. This is another zoonotic disease, meaning it was transferred from animals. And in this case, bats were the original source populations, but there's an intermediate where camels were that transfer to humans from the local bat populations. Once in humans, it was able to spread between individuals. Through respiratory droplets. The COVID-19 pandemic resulted in increased research into vaccination technology. While the concepts used in the new mRNA vaccine have been tested and established for years, the implementation of the new mRNA vaccines were novel and effective. Now, is COVID-19 the first pandemic that the United States has experienced? The answer is no. While the United States was not severely impacted by the SARS and MERS outbreaks, in the early parts of this century, that does not mean that we have not experienced pandemics before. The 1918 Spanish influenza pandemic was very severe. Do we think COVID-19 will be the last pandemic experienced by the United States? And the answer is, unfortunately, no. There will certainly be future pandemics in the United States. In fact, globally, pandemics are increasing in frequency due to increased human-animal interaction and through the globalization of travel. As humans like to spread to new environments, viruses that encounter those humans will then spread to those environments as well. So where did SARS-CoV-2 come from? Well, it is believed that SARS-CoV-2 is a zoonotic virus. and it is thought to be associated with bats, but it is still not entirely clear what the secondary host was, which bridged between wild bats and humans. Throughout the pandemic, we have seen the formation of new variants of concern in SARS-CoV-2. These variants would originate in certain regions, influence the makeup of the local virus distribution, then usually spread throughout the globe, impacting each community, but often on slightly different schedules. So what makes a virus more problematic? Well, from the perspective of public health, viruses become more problematic if they can spread more easily, cause an increase in symptom severity, or do not respond to previously effective treatment. If vaccination or past infection does not give you protection against getting this new variant, we often call that immune escape. It has escaped the defenses that the immune systems have established. So when we look at variants to the SARS-CoV-2 strain, after the initial strain, the variants were named based on letters of the Greek alphabet, with the first variant being referred to as alpha, and then beta, gamma, delta, and then Omicron. Now, Delta was recognized as causing more severe disease, while Omicron was more contagious and would even evade the immune system of the vaccinated and the previously infected. And at the time of this recording, which is in the summer of 2022, there have been many substrains of Omicron, and those are still the most prevalent strains of COVID-19. In order to track these changes in the makeup of the COVID virus frequency, it required sequencing. To know which strains of COVID are in an area and which strains people are getting, an infected person, a sample of the virus would be taken from them, and the genetic material would be analyzed. And it would identify which COVID variant was surging in a particular population. So, what do we do? What are our next steps? How do we continue with this? Well, testing and viral typing are important tools to help preserve public health during a pandemic. We will hopefully be better equipped to deal with future pandemics by what we are learning through this COVID-19 pandemic. And public behavior and response plays an important role in controlling pandemics. So what can we learn from this? Well, first off, effective communication. And plans from our government leaders will be important when we experience future pandemics. Regular testing, tracking, and vaccination research should also continue. Amplifying quality and reliable sources of information is a role that the media can play, and having a clearer understanding within the populace of what is to be expected and what the next steps might be. These are all ways for us to deal with accurate, correct information when dealing with a public health emergency like the COVID-19 pandemic. In our next module, we'll be talking about cell division and cancer.